The overall goal of this demonstration is to compare 3 different cell proliferation assays and present the advantages and disadvantages of each method.
This method can help answer key questions in the cancer research field, such as the use of the most appropriate cell proliferation method.
Demonstration of this method is critical as the cell imager steps are difficult to learn. They require you to focus on the cells before applying the appropriate cell counting mask.
To begin this procedure, grow 2 MCF7 cell lines for 3 days to 75 to 80% confluence in the T75 square centimeter tissue culture flasks using phenol red-free DMEM. After 3 days, remove the cells from the flasks by pouring off the medium into a waste container. Then, immediately wash the cell layer with 2 milliliters of pre-warmed 2x trypsin. Aspirate the trypsin and add another 2 milliliters of pre-warmed trypsin to the cell layer before placing it in an incubator. Once the cells are detached, wash them with 10 milliliters of warmed, fresh supplemented DMEM. Then, transfer the cell suspension to a sterile 15 milliliter tube and centrifuge it for 5 minutes at room temperature. After 5 minutes, carefully aspirate and dispose of the supernatant. Afterward, resuspend the cell pellet in 5 milliliters of fresh supplemented DMEM. To perform a cell count, dilute 100 microliters of the cell suspension